And so, Jonathan, if you want to start, the floor is yours. Thank you very much.
Thanks a lot, Camille. Thanks a lot, Nicolas, for the introduction, and thank you for hosting us as a group at Cesab. It was a very great experience for us, and I try to show you some of the results of the group. So, hello everyone, good afternoon. So I'll just try to change the slide. Yeah, it works. So today and this afternoon, I'm going to present you some of the output of the working group on global redistribution of biodiversity through a macro and eco-evolutionary approach. First of all, a quick overview of the participants of the Bioshift group. We started maybe by being a bit more than 12 people at the very beginning, the first meeting, and then we expanded a bit the group. And so now we are about 25 members in the Bioshift team. It includes a lot of early career researchers within the group, as well as some colleagues from the US Geological Survey. And now I start by giving you a brief introduction of the context of the Bioshift working group. So the context is about isotherms on the move. So what do I mean by isotherms on the move? Isotherms are those invisible lines that you cannot see, but they are just the point that connects an equal quantity of heat on a map. So for instance, mineral temperature or maximum temperature of the warmest month as the two example variables that you can use to depict those isotherms. And isotherms, since a couple of decades now, have been showing some moves in latitude and elevation. So for instance, poleward shift of isotherms is now demonstrated in Europe. There is a paper from Martin Beniston in 2014 showing that European isotherms have been moving 50 kilometers per year on average using climate analogs. And similarly, in elevation and in mountain ecosystems, the isotherms are moving up slopes. So upward along the mountain slopes, as you can see on this picture of the Matterhorn in Switzerland, where you have actually different isotherm distribution depending on the northern versus southern slope of this iconic mountain in Switzerland. And of course, those isotherms that are moving up slopes and poleward, they affect the distribution of ice on Earth and also the biodiversity. And here is just an example of the consequences that are visible. So this is probably the tip of the iceberg in terms of consequences of isotherms on the move. But this is a clear and visible consequence. So the shift and the retreat of glacier over time. Here is the example of the glacier from Argentière in France, with a picture from 1890 and now a picture of 2015. And you can see the drastic changes and the drastic shift in the glacier retreat, where in the past, basically, the people living in Argentière could touch the glacier right from the outside of the village, and now they have to climb, several hundred meters upslopes to actually touch this glacier. So there is a drastic change also in the distribution of life, as you can see here with the trees that have been expanded upslope following the retreat of the glacier. But then the question is how fast the biodiversity is moving upslope or poleward, and does biodiversity track the velocity at which those isotherms are moving in space and time? And so there is already a lot of literature on biodiversity redistribution, so species moving poleward and upslope. And now, and this is of course the main topic of the Bioshift working group. And with this overall objective of the group is to actually gain a better understanding and predictability of species distribution in response to climate change. And so we try to use an approach that combines intrinsic factors. So intrinsic factors are basically species traits and macroevolutionary processes that are internal to the species characteristics, so the evolutionary history. And then extrinsic factors, what we call extrinsic factors, are, for instance, the climate change exposure. So how fast the climate is changing. The climate change velocity, for instance, is one metric used to assess climate change exposure, habitat fragmentation, land use change in general, and human pressures. All these extrinsic factors will affect the distribution of species. And so the combination of those two, extrinsic and intrinsic factors, sometimes interacting in complex ways, may explain the velocity at which species are responding and shifting their distribution, together with biotic interaction, which is an important component to understand the velocity of species shift. So that's the overall framework we are working on with the Bioshift working group. And to answer some more specific questions, I will just list a few of them. I'm not going to respond to all these questions through the 40 minutes presentation, but that's to give you an overview of the project aims and research questions. So first, how to best capture climate change exposure using climate change velocity metrics, thermal sensitivity for intrinsic species characteristics, how habitat structure interact with species traits to explain the velocity of species shifts, so habitat fragmentation, for instance, and dispersal capacity for species intrinsic traits. Can the non-climatic factors mediate species responses, such as land use change or fishing pressure in marine ecosystem? And can the macroevolution aspect, so macroevolutionary processes, inform adaptive potential? So using genetic diversity or phylogenetic signal. So I will touch upon a few of those things during the presentation, but of course not all aspects. So first of all, what we did during the 3 and almost 4 years of the project was to first focus on the conceptual development, publishing a kind of opinion paper on how we can bring back the traits into the equation to understand species shift. Because in the scientific literature, as you may all know, there is a lot of debate on whether the traits of species really can explain the velocity at which species are moving in space in response to climate change. And so the few meta-analyses and syntheses that have been published so far on this topic or this research question have demonstrated that traits are poor explanatory power to explain the velocity and direction of species range. So what we did is to try to propose also to carefully integrate some aspects that are sometimes not incorporated in those meta-analyses, which is, for instance, accounting for methodological attributes, or what we call here estimation processes. So the type of data used, the spatial characteristics of the study where species shift have been reported, the temporal aspect and the temporal characteristics of the reports of species shift, as well as the statistical method used to assess the velocity at which species are shifting their distribution at the trailing edge or at the leading edge of the distribution. All of these will affect and bring some noise in the variability of the reports of species shift. And so we need to account for this kind of methodological attributes to better understand the impact of traits on the velocity of species shift. And so not only we need to account for those methodological attributes, but also for complex interactions between the species traits themselves, so which is here depicted in the ecological processes and the number 2 box here, with like ecological generalism, physiology, phenology and behavior, biotic interaction, species dispersal capacity, and the lifespan, for instance. So all these kind of categories of traits can, of course, explain potentially the velocity at which species are shifting, but they may interact in complex ways with extrinsic factors such as the exposure to climate change, habitat fragmentation, and other biotic dependencies between species. So if we do not account for this complex interaction, we may miss or we may not be able to explain with traits the velocity at which species are shifting. That's what we wanted and intended to say in this opinion paper. And to demonstrate a bit that, we use simulations with this idea. So this is just here a quick illustration of what we meant by accounting for complex interaction between the velocity at which isotherms or climate change is changing and the traits themselves. So for instance, if you wanted to look at the relationship between a given trait, like maximum dispersal distance or species thermal tolerance, and how this trait can explain the velocity at which species are shifting in terms of meter per year in elevation or kilometer per year in latitude, so the y-axis here on those two plots, and the x-axis is a given trait. And so in those two plots, you can see a big cloud of points. We can here more or less guess a positive relationship for the first trait and a negative relationship for the second trait. but the explanatory power is going to be poor given the huge variation in the data points. But in fact, if we incorporate a third dimension, which is how fast, I'm sorry, my phone in mute mode, sorry for that. So if we try to illuminate this relationship with also the velocity at which the isotherms are moving in space, here it could look like this. This is just, of course, fake data. I'm just showing you what I wanted to explain in terms of complex interaction. You can see here that if you do not account for the velocity at which isotherms are shifting in space, then you may miss a lot of the information. Because if the velocity of isotherm shift is very fast, so the velocity of climate change is fast, it's high exposure to climate change, then you could here expect that the link, the positive link and relationship between maximum dispersal distance and the velocity at which species are able to shift is much stronger than if you have a low velocity of isotherm shift. That's what we meant here by this complex interaction between the exposure to climate change and the traits themselves. Same for thermal tolerance. You could have a situation where under a lower velocity of climate change, the link is quite poor between the velocity of run shift and the thermal tolerance. But then as the velocity of isotherm shift increases and you have higher exposure to climate change, we could expect, for instance, just hypothesis that species with a higher thermal tolerance are likely to shift fast and at greater speed poleward or upslope, for instance. So this is just an example, but we did kind of simulation using a simulation exercise with several traits and different velocity of climate change. And we could explain with this simple simulation that if you incorporate those complex interactions between traits and the exposure to climate change in terms of climate change velocity, you could get a lot of explanatory power and go from 33% of explanatory power with just a single trait to 99% here by incorporating the complex interaction between the trait, maximum dispersal distance, and the velocity at which the climate is shifting. So that's kind of the message of this first conceptual paper. Then here I'm going to present more insight in terms of the data we used during our 3 years within the Bioshift group. So we focused on the Bioshift database that we published just before we started the group meetings. So in 2020, we published the first version of the Bioshift database that is available, freely available on Figshare, still available there actually. But during the 3 years, we also consolidated this database with another one that was published by the US Geological Survey and by Sarah and Madeleine, who came to this core database, which is a contraction or range expansion database, which also gathered information similar to Bioshift, but using more, actually retrieving more data in terms of qualitative information for range shift studies. So qualitative shifts were also incorporated in this database. There was also more recent articles than in the Bioshift database. And when we tried to combine those two databases together, we ended up with some mismatches and some issues. And so we had to fix it and to find ways to combine and to get a consensus version of Bioshift plus core. And that took us some time to get to this point. And now we have a new version of the Bioshift database that is still not yet published, but we hope to publish at some point anytime soon, but we'll see. So this is just here a quick representation, and this work is led by Jake Lawler, Nikki Moore, and Jennifer Sunday, together with Liz, me, and other people from the US Geological Survey, where we try to now combine all this data from core and Bioshift in this unified version, where you have the run shift data at the core of the database that is linked to taxonomic information as article attributes, because run shift data come from studies that are published in the scientific literature. And so there is metadata that is at the level of the article or the publication. Then there is also study area polygon database. This is just due to the fact that we tried to get spatial information from the run shift data by digitizing the study polygon of the study area covered by the different publication and reports on run shifts. And so we have study polygon attributes that are related to those polygons. And now we are also trying to refine those polygons with species relevant polygon database, which is for each study area. For example, here we have the case of a study in Northern America with one specific species that has been studied in this study, specific study, which is the Acer rubrum, so red maple. And as you can see here, we have the study polygon of the study area in black, but we also try to get a species-specific subset of those polygons by cropping or intersecting these maps together with range maps from the specific species that has been studied by those authors who reported run shift here for Acer rubrum. So this kind of refinement takes time, but this is a new version of Bioshift that is under development. And so there will be also a companion R package together with this new version of Bioshift, the Bioshift R package that is also under development. And this is Jake Lawler who is working and leading this task on the Bioshift R package. And you can already have a look on it in a GitHub version, but this is still under development and work in progress. From this database, what we did also within the group is to publish a review paper where we try to show how much the content of Bioshift is very heterogeneous in terms of the magnitude of the shift themselves, but also in the direction of the shift. So when we looked at the database, actually the first version of Bioshift, there is a substantial amount of variability with only 60% more or less of the shifts that are consistent with the temperature-based expectation, more or less 60% of the shifts that are shifting according to the isotherms shift. So there is quite a lot of variation and up to 40% of the shifts that are not really according to isotherm shift, with some species shifting equatorward or downslope in elevation. And so this variation is quite important in this database. So there are other, of course, predictors or variables that could explain this variability besides temperature and isotherm shift that I was mentioning at the very beginning of the presentation. For instance, habitat fragmentation or habitat limitation can limit the velocity at which species can move, or sometimes go in opposite direction if you have like habitat fragmentation that is happening at the leading edge of the range. You could have also effects that are due to methodological attributes, so data issues or detection methods that may also contribute to this variability in the range shift detection. There is also possible biotic interaction, climate driver other than temperature, such as precipitation, for instance. Precipitation changes can also reverse the expectation of species run shift. There is examples of studies that have reported species shifting downslope to actually track the change in precipitation in elevation, for instance. And there is also, of course, dispersal constraints that could explain those unexpected run shifts. So there is a lot of reasons to explain those shifts that are not tracking isotherms. And there is also a lot of variation in the Bioshift database itself. As you can see here, I've just represented the taxonomic and the geographical bias. So the geographical bias is represented here on the right, on the left, sorry, with Europe being together with North America the two continents where we have most data of species run shift, with very few information from Asia, Africa, Oceania, South America, or Antarctica. And a lot of taxonomic bias too, with a lot of run shift reports for birds or vascular plants in general. Also for terrestrial and freshwater arthropods in Europe. But for the other taxonomic groups, we have much less data. And also the database is slightly biased toward terrestrial ecosystems, with more reports in terrestrial ecosystems than in marine ecosystems. So that's all the thing we have to bear in mind, and you have to bear in mind about the results I'm going to show you afterwards. There is a lot of variation in the database itself, and a lot of heterogeneity in terms of a lot of bias in terms of taxonomic and geographical coverage. One output that is still unpublished but still ongoing work, and we actually resubmitted this very recently. This work is led by Bruno Oliveira, who is the postdoc and the main leader of this work within the Bioshifts working group. So Bruno has been leading this study, which is about trying to explain the observed velocity of species range shift by going beyond the simple isotherm shift expectation. So what we did here is to build a species distribution models to actually try to model the expected shift if the species would track their climatic niche. So not only temperature, but other bioclimatic variables such as the precipitation variables or other temperature-related variables. So we combine a lot of bioclimatic variables in a species distribution modeling exercise to model what would be the shift if species were shifting according to their climatic requirements. And to do that, we combine the documented redshift from Bioshifts, the database itself. So from the scientific publication, we extracted the latitudinal redshift. So for this study, we focused only on latitudinal redshift, not elevational redshift, for several reasons, but we focus only on latitudinal redshift here. We use those study area polygon of each publication to try to then assess with SDMs how fast the species should have been moving within the study area of interest from the study itself and over the study period that the study area is covering. So for each study level data, we actually try to reproduce all what the authors have been reporting in terms of how long the monitoring has been done for reporting a shift, and also over which study area it has been reported. And then also we recomputed the shift of the range position. So for instance, maybe in a given publication, the authors reported a shift of a butterfly species in France, with shift at the trailing edge, the leading edge, and the centroid of the range. They reported a shift of 2.5 km per year at the leading edge, 2 km per year at the centroid, and 1 km per year at the trailing edge. There is specific information on the metadata for this specific study. We use this metadata to reproduce the shift for this target species, this butterfly species, by using the same starting year, the same ending year, and then predicting the climatic envelope of the species across the study area from the beginning of the study period to the end at a yearly basis. So we have yearly time series of SDMs covering the study area using species occurrence data from JB, for instance, or other information or databases on species distribution, the raw occurrence data. And then with those model range shifts, we track the position of the leading edge, the trailing edge, and the range centroid, as the authors did in their report of species range shift. And so we were able, with this time series at a yearly resolution from the beginning to the end of the study period of the focal study or focal publication, we could reproduce or recompute expected range shift from following the exact requirement of the authors. And so we try then to match or to see how the predicted shift in latitude, in velocity, so kilometer per year poleward, are matching with the observed one. So the model latitudinal range shift, how they match with the study reports for this specific study and this specific species. Here you have, for instance, we were tracking the leading edge by simply computing the slope of the relationship between the leading edge position over time using the time series of the SDM. So using this kind of information, we did it for a set of 500 or so marine species. So we fitted SDMs for 500 species of marine ecosystems and about 3,000 species from terrestrial systems. And this is more or less the distribution and the number of species per spatial unit that we covered from the Bioshifts database in terms of latitudinal range shift. So from this set of species, where we could do SDM exercise, we then tried to answer several questions. So we computed the expected range shift from the SDM's prediction, as I was just explaining to you before. And then we related the model range shift against the documented one. And as you can imagine, well, we could expect sometimes that it was aligned. So what we call alignment is that if the study, if the authors report a shift that is poleward, then we may report also poleward range shift, and so it's aligned. Or the opposite, we could report a negative shift in latitude, so equatorward, and the same for the authors. Maybe they reported in the original study equatorward shift, so a negative shift over time. And so we have alignment in purple colors here. And in yellow, this is a misalignment when we actually predict opposite direction to what is observed by the original authors. So we actually quantified the amount of shifts that are aligned and misaligned first. And then we looked at the magnitude of the coupling, so how much they are correlated, those model latitudinal range shift with the documented ones, expecting that if the SDMs are good, they would be positively correlated and with a linear relationship and a slope that is close to 1, if it's perfect. But of course, you will go in, you will see that it's not perfect. And then we also try to explain in the third question what are the driver of the mismatch, so the difference between the predicted shift and the observed shift. So we computed the mismatch as the absolute value of documented minus model shift in latitude. And this mismatch in absolute values was categorized in 3 different levels. The overshoots. So overshoots is simply when the model is predicting a shift that is faster than when the documented shift is faster than the model shift. So species are shifting faster than what we would expect from climatic envelopes and SDMs. The lagging type is when the shifts are the document shifts are smaller than the expected one from the SDMs, and the misalignment again is when we predict opposite to what we observe. And so we try to explain for those 3 categories of shift, overshoot, lag, and misalignment, what are the possible drivers of the mismatch in terms of model performances, so the SDM performance, landscape connectivity within the study area, is it a habitat that is very fragmented or not, the variability in the suitability of habitat species distribution models over time. So using the SDM time series, we could actually compute whether these suitability values are very fluctuating over time or very stable, expecting that if it's highly fluctuating, then it might be very different from a very stable change over time. I mean, a smoother change over time. The sampling methods and the methodological attributes, and then locomotion mode, for instance, the dispersal abilities of the species. We try to thus use those extrinsic and intrinsic as well as ecological processes or methodological attributes to explain, those were used to explain the mismatch. And the results here, the main output of this study is that we found, of course, a poor match between a model latitudinal range shift and documented one. So the model one are in the x-axis, the observed one are on the y-axis. This is again only latitudinal range shift for terrestrial on the left and for marine on the right. And you see the correlation is pretty poor. There is a very low correlation. And we have also here on this inset the number or the percentage of points that are aligned in purple and misaligned in yellow. And as you can see, the alignment is better for marine ecosystems than for terrestrial ecosystems, but still we have a huge variability and a poor correlation between observed and model range shift. So we then computed for all those taxonomy groups, so ectotherms, endotherms, cryptogams, and phanerogams, so the plants. We then extracted this difference between modeled and documented latitudinal range shift, so the mismatch in absolute value with the 3 different types of mismatch, as I was explaining before: misalignment, lagging, and overshoots. And so here is just the results for terrestrial species, so about 3,000 species in total for terrestrial systems. And for those 3,000 species, we can see that the mismatch in terms of difference between prediction and observed redshift is slower or lower when we increase the model performance. So this is an expectation we had. We expected that the better the SDM, so if we had a better SDM performance in terms of, for instance, AUC, but here we use the continuous Boyce index as a measure of model performance. And so the Boyce index is closer, when the Boyce index is closer to 1, so very good SDM performance, then we have a low mismatch. So the model performs better and the expected redshift are matching with the observed redshift pretty well. So that's good news, actually. It shows that SDMs can be informative, but only if you have a very good performance, at least for the lag group, so the group of mismatch that correspond to lagging dynamics. For the other two groups, the relationship is not significant. And then for the study duration, we have a negative effect. So the longer the study duration, the lower the mismatch. It's also somehow a bit expected that if you have a longer monitoring, then there is a lower mismatch between expected or model redshift and observed redshift. The latitudinal extent also has an effect, but positive this time for all three categories of mismatch. So the greater the latitudinal extent, the greater the mismatch. So when you focus on a very large study area, then it's more likely that we find a mismatch between the model and expected shift. It's kind of obvious, but it's good to show it that, of course, if you focus on a larger area, you are more likely to find a mismatch than a match. And then we also find an effect of the variability, actually a very strong effect of the variability in terms of suitability values over time. So when it's very extremely variable over time, then you have a lower mismatch, higher mismatch, and the mismatch decreases and is lower when you have a more stable condition over time. So that's for this study. Then we had we have another study that is still under revision, also led by Bruno, the postdoc, and together with Jonathan Roland in Toulouse. And so this specific study is about the genetic diversity and how genetic diversity can help to explain the velocity of species redshift. So we had a bunch of hypotheses for explaining, again, latitudinal redshift at the leading edge or the trailing edge with genetic diversity, expecting that genetic diversity, higher genetic diversity should favor higher shift at the leading edge, while it would also favor lower shift or no shift at all at the trailing edge. So that's the expected assumption at the beginning of the study. Here again, we use only species that are showing latitudinal redshift in the Bioshifts database. So the data from Bioshifts are below, the map is below. And the top map is another map from the Fonseca database on genetic diversity metrics. So we retrieve information on genetic diversity for several taxa using the database from Fonseca and colleagues published in 2023. You have the number of species per spatial units here in log scale for both databases. And when we combine these two databases, we were able to focus on a set of almost 2,000 species from 7 major taxonomic groups: insects, birds, fishes, plants, and arachnids, covering mostly terrestrial, but also a few marine species, if I remember. And we built a model where we tried again to explain the absolute value of the latitudinal redshift. So how fast the shift is in terms of absolute value. And we try to explain this velocity of redshift in latitude together with data from the Bioshifts database, such as the position of the so at which position this redshift was reported. Is it the centroid, the trailing edge, or the leading edge? The genetic diversity. JD is for genetic diversity, so intraspecific nucleotide diversity. The velocity of isotherm shift also in absolute value, so how fast isotherms are shifting forward for the same study area here. We had tested for a complex interaction between genetic diversity and the velocity of isotherm shift. And we also added several covariates that are methodological attributes, such as sampling design, data type, study area extent, and grain size, together with also a random effect of the taxonomic class, for which we have reports on OneShift. So we use this model, it's a linear mixed effect model, to try to see if we could find a relationship or an effect of genetic diversity to explain the velocity at which species are shifting. And we found a negative effect of genetic diversity on the absolute velocity of species run shifts under high exposure to climate change. So this was an interesting result, actually. So for all range position, whatever the position, there is a negative effect of genetic diversity for very high climate change velocity. So you see this interaction here with climate change velocity. Again, like for the trait I was mentioning at the very beginning, it's again very important to account for complex interaction between species intrinsic characteristics, such as genetic diversity, and the velocity or the climate change exposure. So climate change exposure here in terms of velocity of isotherms is higher with the red colors, so 7 km per year of isotherm shift here. And for this very high and high-speed velocities of climate change, you see that genetic diversity tend to decrease the velocity at which species are shifting their range to track those isotherms. So that was a kind of interesting pattern. And at the leading edge, we found another interesting pattern at the leading edge. Under low climate change velocities, the genetic diversity this time has a positive effect. So higher genetic diversity tend to favor a faster shift at the leading edge, but only if climate change velocity was not too fast and kind of slow. So it's kind of related to the velocity at which the species is exposed to climate change. Another work that was led by the US Geological Survey and by Laura Thompson from the US Geological Survey is about adopting the Resist Accept Direct Management Framework. So the RAD framework is a very famous framework for in terms of conservation and biology under climate change. And using the RAD framework, how we can somehow adapt this RAD framework to the context of species redistribution under climate change. So looking at the resist actions basically we accept the change in terms of friendship species, whereas the resist strategy will try to allocate effort to allow the species to persist at the trailing edge, for instance. So the RAD framework was kind of interesting to discuss in light of the species redistribution context. So now a few feedback. I'm just looking at the clock here. So I'm soon at 40 minutes. So I'm just finishing with some feedback from our group in terms of project coordination. It's just a few bullet points. It's not exhaustive at all, but trying to give you some feedback on this. So I think first, what we wanted to say is that identifying very early during the project the lead authors to different set of target papers is quite important, and it can help to achieve those targets. So this is important to right from the start identify the lead authors and not only rely just on the postdoc that is working within the working group. Of course, it's a group thing, and it's best to have several lead authors identified early on during the project. It really helps. That's something we realize. Working remotely to supervise Bruno, for instance, in our case, was not always easy. This is a drawback of CESAB. It's really nice, but when we are not like on site, it's more maybe difficult for PIs of the working group to really help the postdoc on a daily basis, one can say. So that's something to do, of course, when you are not in Montpellier and the postdoc is based at the CESAB in Montpellier. it's important to plan those exchange with the postdoc for supervision because it can be challenging, and it's good to plan ahead the research visit and benefit from this very frequent visit in Montpellier. The 6 months meeting really is a nice thing to interact with the postdoc, but yeah, it's still remote if you are not based in Montpellier, so it can be challenging. That's the point. Involving also the recruited postdoc in the scientific animation of the working group is something I think very important, because it's a win-win situation also for the PIs that can benefit help from the postdoc, but also for the postdoc to really be involved right from the start in terms of scientific animation. And it's also good for the CV for early career researchers. So I think it's another point that would be, that is nice actually to consider. Identifying a third person in the core group, because most synthesis working group have 2 PIs, it's kind of good also to have a third person just as a backup, just in case one of the 2 co-PI is not available or cannot attend one of the meetings and so on. So it's not a luxury to identify early on during the project a third person that is willing to invest more time and more effort than maybe most regular participants. So besides the 2 co-PI, have a third person in mind, it's good. Sorry, I'm going too fast. Planning of writing a group session also, that's good during the working group meetings. For instance, really dedicated to writing a specific paragraph or section of a paper. We realized that it was really helpful to get things done on a given paper, especially, of course, not during the very first meetings, but towards the late meetings. We realized that it was nice to have writing sessions by separate groups, little working groups that were focusing on writing. Other feedback, yeah, about targeting more specific subgroups to advance on particular tasks during the working meetings to avoid dispersion. And sometimes we wanted to tackle too many things during one single meeting, and so we realized that it was too much. And so sometimes it's better to maybe not over-plan or want to do too many things at the same time. So maybe for a given specific workshop, it's good to be more focused sometimes to get really the things done, because if you want to do too many things at the same time, then you have this impression that you have done nothing, which is not the case, but that's the feeling you can get if you want to tackle too many things in parallel. One important thing is to consider very seriously the data management plan. It's not mandatory, but it's now more and more used to plan research, to also have a data management plan. And I think it's good to also use this kind of data management plan for the working group at CESAB. Because sharing data and code in a common repository is one thing, but organizing the data and the code is very important for the group and for reproducibility. So this is something that you can really benefit at CESAB. There is a lot of tools and a lot of support to help you organize and share your code and your data in a common repository. That's something we have learned a lot together with Nicolas Casayus in the group. So it was really helpful to have Nicolas helping us on that. And this is super nice to think about data management plan ahead. Also, together with this data management plan, it's good to plan for big storage needs and big computer power. In our case, we did quite a lot of computer power, and so it was important to consider like big computation platforms. So if you need that, you have to plan it quite early on during the project, and then planning ahead for yeah, that was just I was saying. So yeah, that's it for the feedback. And then just to finish this last slide about a few take-home messages from the main results I was showing you today. So species on shift velocities do not conform to expectations from climate change, as you could see with the SDM exercise. So a lot of species are actually shifting differently from what we could expect, even from SDM that incorporate more than just temperature and isotherm shift. So there is more complicated stuff than just climate change as the main drivers. Methodological attributes need to be accounted for to understand the wide variation in on-shift velocities. This is something that I've been trying to insist on throughout the different slides. So we always incorporate this methodological attribute to account for the noise due to different methods used to assess run shifts. Climate change velocities interact in complex ways with species in characteristics or intrinsic characteristics, so such as trait or genetic diversity, as I was just showing to you with the last slide with genetic diversity. Genetic diversity fosters range expansion at the leading edge, but only under low climate change velocity, under low climate change exposure. And genetic diversity promotes species persistence at the trailing edge under high climate change exposure. So that's the main feedback from the use of genetic diversity to get insights from species run shift, from our understanding of the species run shift. And the future perspective we have for the Bioshift group is to, of course, finish our database development to incorporate also species trait in a more meaningful way, especially for cross-taxonomy group comparisons. It's not always easy to have a common trait to compare birds with fish, with plants, and so on, as you can imagine. So this is a big challenge. Develop a web user interface or Shiny app to allow users interact with the database content and maybe, if possible, feed it with new data on run shifts. That's something for a long-term target, but we are thinking about it. Match the Bioshift database with biotime and data on trophic networks and non-trophic biotic interactions to better understand also biotic interaction, how they may matter for understanding run shift. And then apply, of course, for more project money to keep working with the Bioshift working group. That's one of our focus at the moment. We have not been successful so far trying NCES and SDIV to keep working on the Bioshift data, but yeah, we have not been successful so far, but we will keep trying. Thanks to all for your attention, and I'm happy to answer a question.
Thank you very much, Jonathan, for this very informative presentation. And so now we have time for questions. So please use the tools to raise your hands, and I will distribute the speech. Jonathan, if you want to stop sharing your screen so that we can try to see each other. Yes, very good. So anybody with a question?
We have a question.
I can read out my question from the chat if that's helpful.
Oh, yeah.
But it's also written down in case I get distracted and ramble. On the 7th slide, there was a graph where you looked at velocity of range shifts at different rates of isotherm shifts, and it looked like at the highest rate of isotherm shifts, the velocity of range shift decreased with lower dispersal distances. And I wondered if we could perhaps look at that slide again, if you could explain why that was, because I think I understood sort of past the inflection point of that graph, but I didn't understand prior to that point.
Yeah, I can try to share again. Oh.
Yeah.
Do you see the right screen? Or maybe not?
Yeah, perfect.
Yeah, it's
Thank
all good
you.
to see it.
That's the right one. Okay. So, well, here again, I'm sorry if maybe I was not clear, but this is a fake data. You know, it's just made it up, right? So it's just to illustrate the point, but this was just a kind of tool exercise for trying to brainstorm on this idea. The actual result of this exercise is in the next slide. Sorry, this is the one. So this is the one we should trust. Maybe I was spending too much time showing this slide and not this slide. But here, when you look at maximum dispersal distance, we didn't find this complex interaction actually. More what happens after. But it was just that maybe we were at some point discussing and expecting a more complex interaction. But what we found out was a bit more simple in terms of interaction. But again, this is simulations. So we just simulated shift based on like simple assumption of a given taxonomy group having this dispersal ability and so on, and then trying to simulate different scenario of climate change, different scenario of dispersal capacity and responses. So you have to be careful. This is all based on simulation. This is clearly fake data, so maybe forget about this one. This is simulation based on like mechanistic model processes, but not really backed up with observation to somehow validate these expectations from model simulation. But this is just to show if we consider this complex interaction, then it can really help understand and help to somehow have trait back in this equation of how much trait can explain runship. Because so far, really in the scientific literature, there is poor explanatory power of traits to explain those runshifts, but maybe because we didn't consider those interactions perfectly in a good way, or we didn't account it for methodological attributes, which I hope I have been able to show you how important it is to understand runship, because it's very highly variable. At least in this database, we are mixing a lot of data and very heterogeneous data. And so if we don't account for this methodological bias or effects, then we are adding too much noise, and we cannot see much in terms of explanatory power from the traits themselves. I don't know if I answered the question, but
thank you very much. No, that helped. Thanks.
thank you, James.
Cool. Nicolas, did you want to ask a question?
Yes, thank you. I mean, thank you, Jonathan. That's a lot of. That's a very long story. A lot of results. I'm particularly puzzled by the results on the genetic diversity, and your hypothesis is that genetic diversity might be a driver of a response, but I mean it's also actually impacted by the environment, and so it might be the other way, and particularly at the age where you expect, very low genetic diversity because very few genotypes actually can manage to cope with the new environmental conditions. So I'd like to get your impression on that.
Yeah, actually, this is something where we discuss a lot at the moment in the group and within the paper in the discussion section. Because here also one important point I didn't mention is that this genetic diversity we could not perfectly match the two databases together at the population level. So it's really at the species level. So we are aggregating genetic diversity from different populations at the species level. So it doesn't really match with some of the shifts that are also at the species level, but sometimes leading edge, trailing edge, and so on. And so, yeah, it's not perfect because it would be better to match the shift and the genetic data at population rather than species level. And then, like you said, I agree that it's maybe more complicated than just a causal effect of genetic diversity on the shift, but more like how the shift could also explain part of the genetic diversity, because it could be a driver of genetic diversity, and the environment could also affect this genetic diversity. So yeah, it's indeed important to consider this, and this is something that we try to discuss a bit in the discussion section of the paper, but it's not yet completely perfect.
Right. I think we have some questions from the CESAB postdocs. Hello.
Hello. Thank you for the presentation. I was just wondering about the different bias you showed in the data, that you had some data from GBIF and from ranges that are concentrated in North America, Europe, and Australia mostly. And do you think the lack of data in the GBIF dataset could also explain the mismatches that are observed in between the range shifts in the literature that could be derived from data that are not on GBIF, and then you modeled based on biased data? I don't know, maybe some are missing in GBIF that are not, and that they could be included in the observed data from the literature.
Yeah, indeed, we tried to, so we use GBIF for fitting the SDMs, mostly it's GBIF data. And so, of course, we had to exclude a lot of species that for which we may have been missing important information from GBIF because they are not very well covered in GBIF, but in the Bioshift database, there is good data for the shift for those species, especially the rare species. So for this group, we excluded quite a lot of species in the end. I was just showing those for which we were able to retrieve enough records of occurrences to fit SDNs. And what we did also, and I did not mention, is that we, when Bruno was fitting the SDMs, that was a bit tricky and very computationally intensive, but Bruno was matching not only the occurrences with the climatic condition in space, but also in time. So for instance, if you had an occurrence in, let's say, 2010, then the climatic condition that was used for fitting the SDM was getting the climatic condition from this period of time, or like within a restricting window. So we try to also match the temporal data to get SDNs that are as best as possible. It's not perfect, but we try to do our best for matching the occurrences with the climatic data. But we could not really deal with the potential bias from GBIF. And I agree that GBIF is very biased. And so you may have a lot of occurrences like near cities or near area where you have a lot of people that are like reporting information, like for instance in Northern Europe or in Europe in general, and in Northern US, but less in the Southern Hemisphere. Also in some area in the Southern Hemisphere, there is a good social activity in terms of citizen science. So yeah, it's really biased, of course, but yeah, we tried to minimize this, but we could not account for the bias in GBIF.
Thank you.
Cool. Does anyone else have another question? I think we might have time for one more, and then I think we can call it a session, but otherwise. All right. Well, then I think we can stop there. Thanks very much. And it's really nice to see all these results that you guys have done and all the parallel projects that were happening. So yeah.
Well,
Bravo.
thanks to the Cesab. Without the Cesab, it would not be possible. So yeah.
Ah, Bruno.
Hey, Bruno. If you wanted to add something, Bruno, for some of the question, I'm not sure I was able to answer everything correctly. So you're welcome if you wanted to add something.
Thank you so much for translating a lot of the work that was built. And just want to say that it was a great pleasure to be part of this group. It's a really group work, and it was a great experience. And I really miss meeting the meetings at CESAB with all the group.
Me too. I missed the last one last year at the same time, approximately. We were going to the Christmas market last year.
Yeah, for sure.
Okay, well, thank you so much. I'm gonna stop the recording.